In general, genetic information flows from DNA to mRNA to protein. This concept is called central dogma by Francis Crick in 1956. Genetic information is transferred from parent to offspring via DNA replication. The process of DNA to mRNA is called transcription. While the process from mRNA to protein is called translation. So, in this video, we are going to discuss about transcription. Transcription is the process where genetic information in DNA is transcribed into an mRNA. The molecule involved in transcription process is RNA polymerase, DNA strand, and mRNA strand. The function of RNA polymerase is an enzyme that links ribonucleotides into a growing RNA chain during transcription based on complementary binding to nucleotides on a DNA template strand. Next, the function of DNA strand is a nucleic acid that carries the genetic information. Next, the function of mRNA strand is a type of RNA synthesized using a DNA template that attached to ribosomes in the cytoplasm and specifies the primary structure of a protein. So, where does transcription process occur? In prokaryotic cell, the transcription occurs in cytoplasm, while in eukaryotic cell, the transcription occurs in the nucleus. Transcription involves three stages. They are initiation, elongation, and termination. So we go to the first stage, which is initiation. In initiation, RNA polymerase binds to initiation site of DNA called promoter site. Transcription factor and crucial promoter DNA sequence called Tata box form the initiation complex at a eukaryotic promoter. At the joint of the attachment, DNA helix unwinds and unzips the DNA strand. The DNA strand of 3' to 5' is used as a template for transcription to form mRNA 5' to 3'. Why we use the DNA strand of 3' to 5'? It is because the RNA polymerase can only add RNA nucleotides to free 3' end. Moving on to the second stage, which is elongation. Elongation is the copy of the bases on the DNA. DNA acts as a template. RNA polymerase adds RNA nucleotide to the 3' end of the growing strand, complementary to the DNA. The RNA molecule is synthesized in the 5' to 3' direction as it continues along the double helix. Next, the third stage is called termination. The process of elongation of mRNA strand proceeds until RNA polymerase reaches a termination site on DNA. In termination, the sequence of bases that marks the termination site signals RNA polymerase to stop adding nucleotides. Then, the mRNA strand and RNA polymerase are released. At the end of termination, both prokaryotic cell and eukaryotic cell will produce mRNA. Eukaryotic mRNA is called primary messenger RNA or pre-mRNA. It must undergo modification or RNA processing to produce a functional mRNA before it exits the nucleus. In the late 1970s, the scientific community has discovered that the coding sequences within many eukaryotic structural genes are separated by DNA sequences that are transcribed but not translated into protein. These intervening sequences that are not translated are called introns, whereas sequences contained in the mature mRNA are called exons. In other words, exons are coding sequences while introns are non-coding sequences. To become a functional mRNA, the pre-mRNA undergoes a process known as RNA splicing. This process is known as RNA splicing because the introns are removed and the remaining exons are connected to each other. Introns are precisely removed from eukaryotic pre-mRNA by a large complex called spliceosome. Spliceosome subunits bind to specific sequences. This binding causes the intron to loop outward, which brings the two exons close together. Spliceosome excites the introns and ligate the exons. This process leads to mature mRNA. After these modifications have been completed, the mRNA leaves the nucleus and enters the cytoplasm where translation occurs. In conclusion, there are three stages in transcription. They are initiation, elongation, and termination. One of DNA strand, which is 3' to 5', is used as a template to produce mRNA strand, which is 5' to 3' direction. In eukaryotes, RNA processing occurs which modified the pre-mRNA to mRNA. The mature mRNA leaves the nucleus to the cytoplasm.